My name is Carlos Lopez, and I'm an assistant professor of biochemistry in Vanderbilt University. So my work tries to understand how biochemical interactions in cells commit the cell to a specific outcome. So cells, the brain of the cell, if you will, is molecules, and these molecules make networks of biochemical reactions, and somehow these networks process signals that the cells feel to give an output to commit the cell to a given fate. So we try to understand how those biochemical networks process information and how they commit a cell to a given fate and how they fail in disease. So the work of my lab is one, understanding network interactions, and then understanding how noise affects those reactions. We actually measure how cells respond to perturbations using many techniques: mass spectrometry, microscopy, single-cell RNA-seq, cell population RNA-seq, flow cytometry. We use a lot of data, and we actually do a lot of work in data integration. And then ultimately we build models that explain behavior, cellular behaviors mechanistically, and then we make predictions, and then we validate those predictions usually with collaborators. So this is where we're different. We use modeling, mathematical modeling. We use computer simulation. We can gather, so right now we can gather tons of data. There's lots of data that people can generate to explore cellular processes, but then what do you do with that data? Because data is not knowledge. You have to extract knowledge from that data. So my lab uses mathematical modeling, computer simulations, and a lot of statistics analysis to extract knowledge and build mechanisms from those big data sets. So initially, when I started as an undergrad, I wanted to be an artist, and my parents wouldn't let me. And so I said, fine, I'll be a theoretical physicist, which is equal and applicable. And I started to do physics, and then I took chemistry, and I really liked chemistry. And then I took biochemistry, and I started to get fascinated by this idea that you have you have molecules, right, that are inanimate, and then at some point they come together and you kind of blink and they become a living organism. And I was fascinated by that. And I've been fascinated by how information crosses scales. And so early on I realized that my interests in biology were more about how do things work at the chemical level. But then to understand the chemical level, you need to understand physics, and then to understand the physics, you need to take into consideration noise. And so that's kind of what drives me, understanding how these things come together to give you living organisms and life processes. And I think the other cool thing is that I found a creative side to science. So we have to be very creative to address complex problems in science, and I think that's what kind of kept me here. I mean, there's noise everywhere, right? We're surrounded by noise, and yet somehow the organization of life kind of comes out. And to me, that's fascinating, that there's so much noise and so much chaos, and yet living systems still operate in this kind of predictable ways, and that to me is fascinating. At the origin of everything, you have atoms and molecules, and we have atoms and molecules at the foundation of every living system. So we need to understand how these things come together. This is why we biochemistry, I think, because in the end it's about biochemical reactions and biochemical processes that give the foundation for the rest of life. So I use this example in my classes often, that for example in cancer, the origin of the disease is a DNA bond that didn't form correctly, and this propagates to proteins, to protein interactions, to the organelle, to the cell. The cell becomes dysfunctional, it affects its whole microenvironment, the organ in the organism, and then next thing you know, you have a person with cancer years later. But the time scale and the space scale of a bond is femtosecond, femtometer, which is 0.00012 zeros and 1 second and meters. And a femtosecond is to a second, like a second is to about a century. So our challenge is really kind of trying to understand how things at the femtosecond, nanosecond, whatever scale affect the organism. And that's the same as trying to understand how this video is going to affect all of life in 100 years. And so this idea of space and time and how information flows across scales is probably at the central challenge that we do in my lab. Network themselves. So one cell might respond differently than another cell to the same perturbation, even if they're sister cells. And I think that these findings will give us a better understanding of cellular processes, and they will let us kind of predict behaviors better. And I think this could have an impact across all areas of basic research, all areas of applied research, from cancer to immunology to biological engineering. I think that a lot of these foundational theories will have a lot of impact across many fields. What attracted me to Vanderbilt was really that Vanderbilt was probably the only place I found that was collaborative in the true sense of the word. People really interacted with each other. They enjoyed interacting with each other. It was not one person trying to pursue their own agenda. It really was a group of scientists coming together to pursue their own goal, and that was really what drove me here, what I was excited about, and I am excited about here. The fact that I can, if I need to talk to an expert in mass spectrometry, I can go, across the street. If I need to talk to an expert in cell biology, I can go downstairs. These are the things I think that make Vanderbilt unique, and that's what keeps me here. Finding a good mentor is one of those things that really can drive your career. Very early on, I had a very good undergraduate mentor at University of Miami. He guided me very well to where I should go, or where he saw my strengths in my career. The same thing for my PhD advisor. He was very good at kind of focusing what I should do. My postdoctoral advisor, I still remember when I was at UT Austin, my advisor said, well, you're not very good at math, but you're really good at biology. Why don't you go for biology? And that was the truth. I mean, I was okay with math, but it was never something that just flowed from me, But I was really good at kind of bringing information together and understanding things from different perspectives. And even though that hurt a little bit because it hurt my ego, it was really good because it opened a lot of opportunities in biology that I would not have pursued otherwise. So mentoring, I think mentoring is key. I did my undergraduate work in chemistry and biochemistry at the University of Miami. And when I was there, I did a lot of work in molecular simulation and quantum mechanics simulations. But then I decided that I wanted to look at kind of bigger picture things. So I went to University of Pennsylvania for my PhD in physical chemistry. And I did membrane biophysics there, trying to understand how membranes, which are important in all cellular systems, how membranes work and what are the dynamics of a network of a membrane. And then after that, I thought I wanted to go more towards kind of understanding the interface of quantum and classical theory in biology. So I went to UT Austin and I worked with understanding how water and proteins interact. And this was very interesting work because it was very fundamental. But I really wanted to kind of answer bigger questions. So rather than pursuing a faculty position, then I went to Harvard Medical School for a research fellowship, where I actually kind of solidified all this background and kind of really became a multidisciplinary scientist, and found cancer as a topic of research that I think brings all this concepts together. So that's how I ended up here.